pair of speakers for today. We have Hinrich Schulenburg, who's a professor of zoology at University of Kiel in Germany, and he's also a fellow of the Max Planck Society, where he's working, where he's running a research group at the Max Planck Institute for Evolutionary Biology in nearby Plön, near to Kiel. He's established this working group on antibiotic resistance evolution. It's really exciting. I was excited to see that. He received his PhD at Cambridge University in 1999. And I first learned of Hinrich and his work through his paper in Evolution Medicine and Public Health, which is the paper that was shared with you on the invitation to attend, the first of the two. And it's a great paper. I really recommend that people check it out. We also have with him Aditi Batra, who is a PhD student at the Max Planck Institute for Evolutionary Biology in Plön, working with Hinrich. Her research focuses on the evolution of resistance to antibiotics and the opportunistic bacterial pathogen Pseudomonas aeruginosa. And she's originally from India, where her first degree I saw was in botany. So it's interesting to see, it was interesting to see your journey in terms of your interest into antibiotic resistance. Okay, with that, I'm going to turn things over to the two of you. Thanks so much for joining us.
Yeah, thanks a lot for the invitation. It's a pleasure to be here, even though it's only virtually. And especially it's a pleasure also to see so many familiar names. Yeah, great to at least be in contact with you via Zoom. We'll talk today about sequential antibiotic therapy as a potent strategy against bacterial pathogens. Aditi and I will share the talk. So in the second part of the talk will be given by Aditi. And before I start, I actually would like to introduce you briefly to the place we are working. And I'd like to advertise the location a little bit, because it has become over the last decade to one of the big hotspots in Germany for evolutionary research, due to an increasing number of PIs working on evolutionary topics at not only at the university, but also independent research institutions like the Max Planck Institute for Evolutionary Biology, but also a marine institute called GEOMA and others. On top of that, we have been able to, and lucky to have a couple of larger research initiatives being funded, and that allows us to be actually very creative in doing evolutionary research. My group itself is in general interested in studying host microbe evolution. I started to do most of my work with the nematode host C. elegans, which we combine with pathogens, and more recently also with its natural microbiome. And there we are developing a couple of different research foci. So we're interested in C. elegans pathogen co-evolution, we're interested in the worm's immunity, natural ecology, and also microbiota interactions that the worm has with various bacteria. As a side story, I began 6 to 8 years ago to also study antibiotic resistance evolution, and now actually half of my group is looking at that. And it still feels a little bit strange still being at the Institute of Zoology, because yeah, some of my colleagues also find it confusing that we're working so much with bacteria, but it's fine. I'm an evolutionary biologist by training. So we're talking about bacterial antibiotic resistance today, and I think most of you are familiar with that, and for that reason, I will keep the introduction short. I think most of you know that we are in the middle of an antibiotic crisis. This was actually one of the main topics of the World Health Organization before Corona. This topic got neglected a little bit, but I think it is sort of recognized again that it's actually not gone, this particular problem and this challenge. And there was a comparatively recent paper in the Lancet that emphasized this point, that there is indeed a dramatic global burden coming from antibiotic resistance. And this was a quite comprehensive internationally led study that elucidated the severity of the widespread rise in antibiotic resistance, and especially multi-drug resistance. So there is at the moment the threat of a post-antibiotic era. And yeah, the core of the problem is, of course, that bacterial pathogens evolve. They can adapt to antibiotics, and that drug resistance is spreading, leading to multi-drug resistance. And if these bacterial infections become no longer treatable, then of course we are moving into a situation that we thought we have left behind us with the introduction of antibiotics. So a couple of suggestions have been proposed now for a couple of years, and this is an excerpt also from a very influential AMR review published a couple of years ago already by the and funded by the UK Wellcome Trust. And of course, the most effective strategy is to use less, because then we're also not selecting for resistance. But of course, there are many incidences where we need to use antibiotics. And so there are recommendations what else we can do. And one of the main suggestions always is to develop new antibiotics. For me as an evolutionary biologist, and I think most of you are also aware of that, it is curious to see that the evolution of pathogens is at least neglected in the development of new measures, if not completely ignored. And this becomes apparent if you do a text search on these influential papers for the term evolution or evolve. So also in the recent Lancet paper, if you do text search on evolution or evolve, the word is missing. It's not part of the paper. The same applies to this influential AMR review. Now you could think perhaps they wanted to avoid the term evolution for whatever reason, but even though the usually used alternative, namely development of resistance and so on, is not part of these papers. So development is used, but only for developing new measures. And of course, this is, in my opinion, a huge problem, because evolution in the end is at the heart of the antibiotic crisis. So if we want to have or develop sustainable solutions, we need to take evolution into account. So this, I think, is trivial for all of you here, but it's still an important point to make, especially when we talk to our other scientists and clinicians and medical people. So the importance of evolution can be nicely illustrated when we look for the spread of resistance in general, and that is highlighted with the graphic that I'm showing you now. So here along the x-axis, you see the years, and then on the top indicated by these lines when a certain antibiotic was first introduced into medical treatment. And here at the bottom, you see an illustration when first resistances were recorded in the clinically relevant pathogens. And in my opinion, there are two important lessons to be learned from this graph. One, resistance always evolves, even if people tell you that they now found the antibiotic against which resistance cannot evolve. That always turned out to be wrong. Second lesson is that very often resistance evolves quite fast. Not always, but very often resistance evolves quite fast. So it is a very dramatic problem and challenge. The very fast evolution of resistance is an additional point to be emphasized, because at least I made the experience when talking to clinicians that they often told me resistance cannot evolve in patients during a treatment. So we actually did a study with cystic fibrosis patients with Pseudomonas aeruginosa infections. We sampled these patients during an antibiotic treatment, and we had a couple of patients that saw a certain antibiotic for the first time. I'm showing you two of these patients, patient 2 and patient 5. Here you have the period of the antibiotic treatment. Day 0 is the time point before antibiotic treatment. And then here we look for resistance of bacteria that we isolated from these patients. We always isolated at least 5 clones of Pseudomonas aeruginosa. And the lines here in blue and here in yellow are the antibiotics that these patients saw for the first time. And you can see that the spread of resistance in these patients happened within days, really rapidly once the antibiotic treatment was started. So this is something that does happen during patient treatment. So what now? So of course we should consider evolutionary concepts to improve therapy. And this is one of the big aims, I think, of all those of you in the audience that are also working on antibiotic resistance. And I think we all believe that by using evolutionary concepts, we can improve therapy, both ensuring eradication of pathogens, but also minimizing the spread of resistance. And I think there are a couple of concepts that are now identified as being quite promising. And today I would like to talk about one of them, which we have now worked on for a couple of years. And the idea to focus on this one and explore it further came up by taking a step back and just thinking about conditions that usually and also generally make it difficult for organisms to adapt to. And this is fluctuating conditions, fluctuating environments. So we know that organisms usually are able to adapt to constant conditions, and this particularly applies to bacteria. So we can find bacteria under extremely high temperatures, extremely low temperatures, high radioactivity, and so on. And antibiotics are often not a big challenge for bacteria. But we also generally know that organisms often have problems if environmental conditions change fast, and also if they change in an unpredictable way. So we thought this is something that we should test in the context of antibiotics. And this is what we did. And our usual approach is that we perform evolution experiments because these allow us to dissect causes and consequences that influence evolution. So we try to use evolution experiments to address the question which treatments minimize adaptation. And we often then combine that with genomics and also functional genetic analysis to obtain a better understanding of the exact targets of selection. And we originally started this kind of work with E. coli, but over the last 5-6 years, we focus on Pseudomonas aeruginosa since it is an important human pathogen. It's an opportunistic human pathogen, but it often comes with multi-drug resistance, and therefore the WHO placed it in the priority 1 group of most problematic human pathogens. And it is particularly problematic in lung infections, but also certain wound infections. Yeah, and we, for addressing the idea of the importance of sequential therapy, we then perform long-term evolution experiments. We usually use the serial transfer approach, and the results that I show you first have been obtained by a former PhD student and then postdoc, Hinrich Schulenburg, who now works at the IST Austria, and still doing some similar kind of work. So we did a serial dilution experiment, and we included 3 different antibiotics with different targets and different mode of action in this particular experiment. And to assess the importance of different kinds of sequential environments, we had 4 main treatment types. As control, we had monotherapies where the bacteria only saw one particular antibiotic, as indicated by the different shades of gray here. Then we had 2 treatments where the antibiotics were alternated in a regular way, either fast or slow. And then we had another set of individual treatments where we alternated these 3 antibiotics in a completely randomized way. And the whole experiment was run over 96 transfers, each transfer lasting 12 hours. And this amounts to approximately 500 generations of the bacteria. And each individual sequence was then replicated 12 times. We then measured a variety of different indicators of treatment success. One was looking for extinction of the populations that were the whole time exposed to sublethal drug concentrations. So the original expectation was that all populations remain alive, but we actually observed that the bacterial populations went extinct throughout the experiment. And what you now see here are the different treatment types, and on the y-axis extinction rate, and we see that there is generally a higher extinction rate in the treatments where antibiotics are alternated fast in a regular way, or where we had random antibiotic sequences. Another outcome we looked at was we looked at biomass increases or growth increases throughout the entire experiment over the 96 transfers. And this is what we then use on the y-axis as a proxy for adaptation rate. So if you get a value of 1, the bacteria were very well able to adapt and cope with this particular treatment. A value of 0 indicates that they have not yet adapted to this treatment. And here we only included populations that survived until the very end. And you see the results in different colors referring to the 4 main treatment types. And if we now look at these kind of results, you can see that there is quite some variation in the outcome and the dynamics. It's interesting that at the end, they all somehow managed and reached a high value, but it's especially at the beginning, and these are time periods similar to antibiotic treatment of patients. So particularly at the beginning, you see that bacteria under monotherapy and slow regular changes, they adapt fast, they're very well able to adapt, but those under fast regular changes and random changes, they don't. They are adapting at a significantly lower rate. So this already looks exciting and promising. So apparently fast changes, random changes can constrain bacterial adaptation. And of course, it is interesting to see and perhaps find out what exactly is causing it. And we tested a variety of parameters, and to keep a long story short, we then found that an inducible physiological effect seems important. And these inducible physiological effects can be measured with a simple experiment where we expose the bacteria for a short time period to one of the antibiotics. Then we completely change the medium and then use a higher dose of the main antibiotic. So again, a short time period only with either, for example, ciprofloxacin or in this case, carbonicillin. We change the medium and then they are exposed to, in this example, gentamicin. And when we do that, then because of the short exposure, the ability of the bacteria to resist the second antibiotic may be altered. And it can either, the cells may either be more protected or they become more sensitive towards the second antibiotic. We termed this an inducible effect. It's like a memory effect. We termed it hysteresis. And if the first exposure results in a protective effect, it would be positive hysteresis. But the more interesting type is, of course, negative hysteresis. So short exposure to one antibiotic increases the killing rate of the second antibiotic. This is what we then termed negative hysteresis. And you see the results here. So here we have the control treatment, no pre-exposure. And here we have a treatment with a short exposure to carbonicillin, and the main treatment in both cases is then to gentamicin. Dead cells are stained in red, and you can see a faster increase in red cells here on the right side when there is negative hysteresis. This is a phenomenon we can quantify, and that is illustrated here. Here you have the time, and here you have the number of viable cells. This is the pre-treatment period, and this is the main treatment with gentamicin. And the two treatments where you had a short exposure to carbonicillin and ciprofloxacin, you see that the killing rate is significantly increased. And we did that for all possible combinations of these 3 antibiotics, and the results are summarized here in a heat map. This is the pre-treatment drug, the main treatment drug. And we see 2 cases, namely pre-exposure to ciprofloxacin or carbonicillin, resulting in increased killing of gentamicin. And we use this information now to calculate a cumulative measure of hysteresis effects across a sequential treatment, across the different switches among antibiotics. And we then found that this cumulative measure is indeed a good predictor of the treatment outcome. And that is highlighted here. So here along the x-axis, we have a measure for this hysteresis effect. And we transferred it or we present it here in a way that here on the right, you have high levels of negative hysteresis. So the inducible effect is particularly strong. And we then correlated it, associated with different measures that we have for treatment success. So these are the number of extinction events. We also measured, and this is what I did not present, multi-drug resistance of the evolved populations and also adaptation rate. And you see that there is a strong association of negative hysteresis and extinction events. And this association is even stronger if we look for multi-drug resistance, so high negative hysteresis leads to significantly lower rates of multi-drug resistance and also significantly lower rates of adaptation. I just very, very briefly touch the genomics that we looked at here, and that actually confirmed that negative hysteresis is important because we identified hysteresis-specific selection of targets. So hysteresis, so the importance of these physiological effects at the switches between antibiotics does induce in selection on its own, and there are responses to the selective pressure. So there are genetic changes that then minimize the level of negative hysteresis. And one factor that we have more closely characterized is a gene that is called CPXS. It is involved in the cell envelope stress response. And here, out of time reasons, not going into details, we are currently characterizing this gene and associated genes in the same operon to better understand how this cell envelope stress response relates to negative hysteresis, to these physiological effects. And with this new data that I'm now not showing, we can indeed prove that this gene is involved in a very specific way in mediating these inducible physiological effects. So the phenomenon of negative hysteresis. What I'd like to show you at the end of my part, and that is a little bit a teaser. So most of what I presented so far is published. I briefly mentioned that we are characterizing the CPXS gene and are very excited to actually better understand how negative hysteresis works, how this inducible effect works. But what we did in parallel is also to ask how widespread is this phenomenon, and is this a phenomenon that we can actually potentially apply for medical treatment. And we approached that in several different ways. And in one way that we followed is that we took Pseudomonas aeruginosa isolates that are genetically distinct. So we have now a panel of 20 Pseudomonas aeruginosa isolates that cover the entire genomic diversity of the species, which we confirmed through genome sequence analysis. And these are isolates that come from patients, but also from environmental samples. And so with this panel, we were able to ask, is the phenomenon of negative hysteresis common, widespread? Does it depend on genotypic background? And the good news is that this phenomenon actually is shown by all of these genetically very distinct strains. So, and these are these distinct strains. And then here you have the treatments. So in this case, the pre-treatment are indicated by these abbreviations here. And these are beta-lactam drugs, the first three, and the fourth one is a fluoroquinolone. And the main treatment in this case was again an aminoglycoside drug, in this case, gentamicin. And whenever you see something in blue, then we actually were able to identify negative hysteresis. If you see it combined with a star, then it's also a significant effect that we could identify with this particular screen that was performed by a new PhD student, Florian Buchholz. And you can see that especially when we start with a beta-lactam, then followed or then switching to an aminoglycoside, we very robustly see negative hysteresis. We then went one step further and also wanted to know whether negative hysteresis also occurs even if the bacteria are resistant. I should add that some of these isolates are actually multi-drug resistant. So we had it covered here, but we then remembered that from another study, we actually had a range of bacteria that are highly resistant against certain drugs. For example, and this is indicated by these abbreviations, we had strains that were, for example, let's say these ones, highly resistant against gentamicin, or highly resistant against beta-lactam drugs. Here it is piperacillin. And even if we now perform experiments with these drugs against which these bacteria are resistant, then the hysteresis effect is still expressed, as you can see here in this graph here. So here the pre-treatment was, for example, piperacillin, and then followed by gentamicin. And you can still see these blue colors for at least many, not all, but many of the cases where the bacteria are actually resistant against piperacillin or resistant against gentamicin. And that for us, of course, looks really, really exciting, because it could mean even if they are resistant to the effect of the drug alone, but the inducible physiological effect is still acting and actually could make the bacteria more sensitive. And so this looks really promising. And this is something that we are now trying to put into the clinic together with colleagues from the clinic to further explore whether this kind of principle can actually be exploited in patient treatment. So this is a teaser. We're working on this, and hopefully we have more exciting results on this topic in the future. And with this, I hand over to Aditi. Aditi, the floor is yours.
Thank you, Hinrich. Okay, so you guys saw right now a sequential treatment involving 3 different antibiotics, which all targeted different components of the cell. Now, these sequences are known as heterogeneous sequences, and they are contrasted to sequences which are homogeneous, which involve antibiotics which target the same component. Now, clinicians often avoid using these sequences because it's expected or assumed that this will result in cross-resistance and therefore treatment failure. But this assumption has never really been tested, and that's what we wanted to do. So, Hinrich, could you go to the next? Yeah, thank you. We had different antibiotics and we made sets from them, and always 3 antibiotics in one set. So we had heterogeneous set in which the antibiotics targeted 3 different components in the cells, and we had homogeneous sets in which the antibiotics targeted the same thing, in our case, the cell wall. With this, we carried out evolution experiments on the design that you see here, which is the same design as before, where we had regular switching treatments and random switching treatment, and also the speed of switching varied. The main readout was extinction, which is what you see here. The open circles represent extinctions caused by heterogeneous sequences, and the filled circles represent extinction caused by homogeneous sequences. And it's already quite obvious that in our setup, at least, the homogeneous sequences performed either as well or even better than heterogeneous sequences. So, the assumption that homogeneous sequences will result in treatment failure doesn't always hold. Now, we were quite interested in this phenotype of extinction, and we focused on how it is distributed across the different treatment groups. The group that we focused on was on this CARD or CEF group. The names are not important, just remember that they all target the cell wall, and they had the highest extinction. That is why they were interesting to us. Now, on the x-axis, you see the different treatment groups, and on the y-axis, you see the different extinction events. And what you can observe is that the fast switching groups, irrespective of the order of switching, always had the highest extinction. So the switching rate in a sequential treatment seemed to be important for us. Now, we had a readout, and we now wanted to see what all different predictors we can use to explain it. So we did look at negative hysteresis, and surprisingly, we actually did find negative hysteresis amongst the different beta-lactams. However, that could not explain the data that we had. So I will not be talking about that today. What I will talk about today is a variation in spontaneous resistance as well as collateral effects. Now, what is spontaneous resistance? Now, bacteria always have this intrinsic ability to become resistant to antibiotics, and the intrinsic rate at which they are resistant to a particular antibiotic is called the rate of spontaneous resistance. This is measured using the classic fluctuation assay, the schematic of which you can see over there. You can have different parallel cultures and plate these out on antibiotic selection plates and get a distribution of mutants. Now, using this distribution, you can calculate the resistance rate of the bacterium to the antibiotic that you tested. Now, using this method, we calculated the resistance rate of all 3 beta-lactams that we had. And the mutants that we got, we then restreaked them to ensure that the resistance was genetic. And the restreaked mutants were then patched onto plates having the other 2 antibiotics to see whether there was cross-resistance. or basically to assess collateral effects. Now, what do I mean when I say collateral effects? When bacteria are resistant to one antibiotic, they can be, of course, resistant to a second antibiotic, and this is what we term as cross-resistance. However, of more interest is the phenomenon of collateral sensitivity. So spontaneous resistance to a drug A can sometimes give higher sensitivity to another drug B, and this can be exploited both in sequential as well as combination therapies. So that's what we tested with our collateral effects. Now, coming to the results of the mutation rate or the resistance rate, this is what we have. On the x-axis, you see the different antibiotics against which the rate was measured, and on the y-axis, you see the rate. Now, the antibiotic DOR or doripenem had the least resistant rate as compared to the other two, which just means that it was much, much harder for the bacteria to become resistant to this antibiotic. What does this mean in terms of our sequential treatment? We use these individual rates to calculate a cumulative spontaneous rate of resistance for the treatment, which is just obtained by multiplying the number of times the antibiotic occurs by the individual rate of resistance on that antibiotic. Now, coming to collateral effects, this is what we found. Now, on the y-axis, you have the antibiotic on which the bacteria were resistant to, also on which the mutants were obtained. And on the x-axis, you have the antibiotic on which resistance or cross-resistance was tested. What is plotted in the heat map is basically the fraction of mutants that were not cross-resistance, which is what we call the fraction sensitive. For example, in the upper left corner, you have mutants that arose on DOR and were not cross-resistant to CAR. And what you can see here is that if you're resistant to CAR or CEF, you're very less likely to be cross-resistant to DOR. So DOR seems to be essential because it has a lower spontaneous rate of resistance, but also a lower rate of cross-resistance. And similar to the spontaneous resistance, we calculated a cumulative rate of cross-resistance for each of the treatments that we had. Now with this, we were armed with our predictors, which were the spontaneous rate of resistance and the collateral effects. And we wanted to see whether they can explain our extinction phenotype. Now, the first graph that you see here is a correlation between the extinction events and the switching rate. And we found that there was a significant correlation, which was also positive. That is, with higher number of switches or higher the switching rate, you had higher extinction events. The second thing that we found was that the correlation between the spontaneous resistance in a treatment with the extinction events was negative. So the lower overall spontaneous resistance that you have in a treatment, the higher the extinction. And lastly, we found that the correlation between the cross-resistance and the extinction events was also negative. And lower the cross-resistance in a treatment, higher the extinction. So to summarize, if you have, Hinrich, can you go back to the previous slide? Thank you. To summarize, if you have higher switching rate in a sequential treatment, more the extinction events and more the efficacy. If you have overall lower spontaneous resistance, higher the extinction, more the efficacy, and overall lower cross-resistance, higher the extinction, more the efficacy. Okay. Now, what do we want you guys to take back from this talk? First of all, evolution lies at the core of the antibiotic crisis. Hinrich, thank you. Evolution lies at the core of the antibiotic crisis. Although you can approach this crisis in many different ways, targeting bacterial evolution has to be central. One way of targeting bacterial evolution is the use of evolution-informed therapy, in which you use concepts of evolutionary biology to try and prevent or delay the rise of antibiotic resistance. One such concept that we have looked at today is called the fast sequential therapy. And the idea behind this is that if you have fluctuating conditions, such as fluctuating antibiotics, it becomes really difficult for the bacteria to adapt to these conditions, since the selection pressure keeps changing each time. And also that you can have certain factors which can be used to optimize the sequential therapy, which include negative hysteresis, which is a physiological interaction which can result in killing more bacteria. the spontaneous resistance rate of the constituent antibiotics of the sequence, and also the collateral sensitivity phenotype between the constituent antibiotics in the treatment. Now, with this, we would like to end. We would like to thank you all for listening. We would like to thank our collaborators and our funding sources, and we would be now very happy to take questions.
Great job. That was a fantastic talk. Thank you so much. We definitely have time for questions. It looks like we already have one hand up. Michael Hochberg, would you like to ask your question? And for those who've posted questions, please consider raising your hand. We'd love to hear you ask the questions, those who posted in the chat. Go ahead, Michael.
All right. Great talk and very interesting results. I think there's a lot of potential here. So it reminds me of a talk I just saw a few days ago with a slightly different mechanism. It was given by Joel Brown at the Moffitt Cancer Institute, and they have what they call extinction therapy. They're looking at what they call extinction therapies, where there are two drugs. One drug is given at the beginning, then a second drug is given at some interval. And the idea is that the second drug should be given reasonably soon after the first, such as be applied during the trough created by the first drug. And that would minimize the emergence of new chemotherapeutic resistant mutations, because it would be a smaller probability of those mutations being present during the trough from the first drug. So I'm just wondering that even though you showed fairly convincingly that there's this negative hysteresis effect, as to whether or not the fast cycling in your results could be explained in part by less availability of these resistant mutations.
Mm-hmm. So I entirely agree that this could be one interesting factor as well, and it is something we have on the agenda, and we also know it would be important to look at that in more detail. So your idea is that you actually reduce population size with the first drug, and then you kill them all off with the second drug.
Right. So the timing is really important. So you want to use the second drug at the minima created by the first drug, so as to minimize the emergence of new resistant variants to the second drug. And if you wait too long, then you get evolutionary rescue and the potential emergence of random mutations, some of which could resist the second drug. And so it could be a contributor to what you're seeing.
No, I entirely agree. And this is so one challenge is we're using the serial dilution protocol, and that is not so controlled regarding the population size. We have developed a protocol and also recently published some work where we do control population size during these transfers. So we have done some first experiments with sequential therapy where we also control population size, but nothing yet in this direction.
Yeah,
so it is really a valid point, and at least for these two studies, we were happy that we could explain the results.
Yeah, so you could easily test it by plating and doing MIC analyses and so forth. Yeah.
No, but good point, Michael.
Thanks.
We have a perhaps related question as well from Carlo Ameli. Carlo, are you still on, and do you want to ask your question or raise any other points related to how this applies to cancer, or how cancer and knowledge of cancer and resistance to chemotherapy applies here?
Yeah, I'm just trying to think about the scaling between this and cancer. So I apologize if I missed it, but I wanted to just hear about, how many generations are happening under each drug in your fast versus slow switching conditions, and what the population size that are evolving, to think about what the scale would be for a cancer patient.
Mm-hmm. So the number across the 96 the number of generations across the 96 transfers is approximately 500 generations.
No, but like for when you have like a fast switch up for under one drug, how many generations before you switch to the next drug?
Yeah, but so we only have the average numbers, and this is as precise as we could get here. And the same is true with the population size. We did not measure precisely in this experiment. And this is, you could argue, a challenge for everyone who's using the serial dilution protocols, because you usually transfer by volume. We did, however, and that relates to my answer to Michael, So we have established a protocol where we can control this. And as I said, so this is, we can apply it to these kinds of treatments. And this is indeed something that is also on the agenda. But I agree with you, this is a very important point to further assess. But again, so we were happy in these two cases because we could explain the results. So we don't think, or at least the largest part is actually most likely explainable by the phenomena that we showed. What I perhaps would like to add also, because that could be of interest to you and Michael as well, I could also imagine that switches among drugs and cancer chemotherapy could potentially also exploit similar kinds of effects. And this is where we also try to join forces with our colleagues here or oncologists here. And at least they tell us that they have some indication that sometimes there can also be inducible physiological effects. That seems to be, so I try to formulate very, very carefully, that seems to be similar to what we call negative hysteresis. So this may be of interest to some of you to also explore, because if you find cases where these inducible physiological effects occur, then that also may be a means of improving cancer therapy.
Yeah, no, we've been thinking about this sort of drug rotations based on the integrated pest management strategies, but I'm just still trying to get a sense. So is it like the 20-minute generation time and you're in the fast condition, you're switching once a day? In the slow condition, you're switching once every 4 days or something, is that right?
So we switch antibiotics every 12 hours in this experiment.
In the fast condition?
Under the fast conditions, yes.
And what about the slow condition?
The slow condition is 48 hours.
Okay. And it's about a 20-minute generation time?
No, a little bit more. So we usually assume like 30 minutes.
Okay. And do you think they hit a carrying capacity before the dilution?
The population size before, Aditi, here you have to help me with the exact numbers.
So we start with around 5 into 10 to the power 6 bacteria per ml. And the population size is not that large towards the end in the first 2 seasons, but after that, it usually does reach carrying capacity. So they adapt quite fast. And we are always, I must disagree with Hinrich, the division time is actually a bit longer than 30 minutes because we're doing it in 96 well plates and not falcon tubes. And so I would go around an hour and a half or something. And then we have 12 hours. So we switch antibiotics. So we switch media every 12 hours. For the fast switching, we always switch the antibiotics as well after 12 hours. But for the slow switching, we only switch it after 48 hours. Okay.
And so because you're in a 96 well plate, you're in a pretty small population size compared to like a patient in a hospital or something. There's the number of bacteria, right?
I must say I'm not super sure because it also then depends upon the amount of bacteria in the particular infection. But yeah, we will likely have like more bacteria.
Okay, cool. Thank you.
But one point to add here, I mean, it is clear for us that we cannot simulate patient conditions in the lab. So these experiments are meant to generate ideas. To identify concepts that could be used. I will never claim that this is now ready for treatment exactly like this.
But we should be thinking about how to scale it up to the therapy treatment. Yeah.
But I really see the importance of what we can do is indeed generate ideas that are not present in medical treatment at all.
Great, thank you.
Okay, we have a question as well from Thomas Cole. Thomas, do you want to ask your question?
Thanks, and also many thanks for the very interesting presentation. I was thinking about the first slides you showed that the evolution of resistance is remarkably fast and will always appear. I remember reading in a publication, which I'm now doing a probably big disservice by making it very much easier than it actually was written. They basically stated that there is a theory that a lot of the mechanisms that we see appearing apparently after using it as a compound are actually already there in the background, because a lot of these compounds come from the environment anyway, or similar molecules. So maybe what we see is not a fast evolution, but in a lot of cases discovery of resistance. And if you allow, there's one add-on question that I thought of because of the transfer topic we were just discussing. which might be very important for clinical implications. How much do you think is this transferable even between bacteria? Would you think you need to re-establish for example, an optimal regimen for a new pathogen? Or is it more that there are organisms that you would predict are reacting the same way, for example, with negative hysteresis? Thanks.
Yeah, these are, I think, two very important questions, and could result in another talk. So very quickly, the presence of the mechanisms. So I think there is usually the belief that an infection in humans coincides with a bottleneck, so that usually we actually have only a few pathogen cells that are entering the bodies. It depends a little bit on the pathogen where the idea is. But from that point, we could argue that you start with a single clone, and if you see actually susceptibility at the beginning of the infection and then rise of resistance, then it is likely to, one possible explanation is de novo, the other one is horizontal gene transfer of resistances that are somewhere in other bacteria of the same person. And that of course can be an important mechanism too. From genome analysis of the patients that we had, we at least have a strong indication that this is really de novo evolution that is happening in patient. But nevertheless, you are likely right that some level of resistance genes could be present, and they often are, because they also are important for other kinds of interactions or resistance to at least certain type of resistance mutations, also relevant for resistance against other abiotic factors. So it is a complex topic to consider, but the point we wanted to make is that this can really happen very fast within a patient. And I think this is an aspect that is usually neglected. Then the second question was about transferability. You have to remind me, what was it?
So maybe this also just to a very naive question. Just if you're looking at a certain bacterial organism, and that I would find it quite interesting. If you find, for example, certain interactions between drugs, what do you think, how easy those can be transferred to other pathogens?
I think this is, again, a very relevant point, and we're also trying to address that by now. So the phenomenon of negative hysteresis, we actually did identify in other gram-negative bacteria, but it's slightly other drugs. We were lucky and happy that we find robustness in negative hysteresis across different genotypes of Pseudomonas aeruginosa. We did something similar regarding collateral sensitivity as studied in the traditional way. And there we actually find much more variation within Pseudomonas aeruginosa, but also if you then compare it to other bacterial pathogens. So I think I think these kinds of analysis are needed to be better able to say whether there is a certain evolution-informed treatment that we can universally apply, or whether this is an evolution-informed treatment that we have to adjust for each individual patient, depending on what kind of pathogens there is. So I think the end result will be a combination of these two aspects.
Okay, thank you. We have next we'll hear from Sylvia, and then we'll hear from Edmund, and that might be all we'll have time for. We'll see. There might be time for one more. So let us know if you want to ask a question. Please, Sylvie.
Yeah, thanks very much. And thanks for that great talk and a great discussion. I think as far as I understand, like what you've done is you've done a monotherapy comparison to randomized treatment, right? And I was wondering whether you've done or whether you're planning on doing comparing this randomized treatment with concurrent treatment of different antibiotics. And I'm just curious, like what you think would happen, like which one would, I think I know what you're going to say, but which one you think would be working better?
Mm-hmm. So I've been dominating the answers. Aditi, do you want to answer?
Yeah, I can take a go. So yes, the combination therapy is commonly used in the clinic, and that's why it forms a major control group aside from just the monotherapy. In these two particular studies, we did not focus on the combination therapy because we were more interested in the sequential therapy and what factors make it tick, basically. However, in the subsequent experiments that we are planning in the lab, combination therapy is always included as a control now. In specific, whether we are testing it against the randomized therapy, no, we do see a effect of temporal irregularity in the beta-lactam treatment. I personally don't see this really translating into the clinic, because you can't be designing really irregular sequences and then giving them to patients. How they would compare to combination is something that we would have to test. A priori, I cannot say what the results would be. I would tend towards combinations doing better, but again, it is something that it's better tested.
My addition to that is that we decided to focus on sequential therapy because it is neglected. So there's actually much more work done on combination therapy. And so we thought it is actually of value to draw attention to this kind of treatment as also a promising and quite potent treatment. We also did not do it in these experiments because doing properly and precisely analyzing combinations of 3 is a challenge on its own. And it would actually be a paper on its, or several papers on its own, because you actually need to properly define how to evaluate the type of interaction among these 3 drugs. And there are a few papers, and they also highlight it's anything but trivial to do that. Because you have to make a decision in which concentrations you would add the 3 drugs to each other. And this actually needs to be evaluated properly, and you would actually have to use a range of drugs of all 3 in all possible combinations. I agree with Aditi. I think this is a very promising approach, and it's also neglected. So most experimental work is done on combinations of 2 drugs, but it's also interesting to see, so the combinations that work, where clinically it is properly proven that they really work, usually consist of 4 drugs, or let's say more than 3 drugs. And I think experimentally there we are lagging behind to actually understand why this is the case and whether there is value in this.
Thanks.
Okay, thank you. Edmund, go ahead.
Yeah, I was very interested in the experiment on negative hysteresis, where just a brief, like 15-minute exposure, non-lethal, had an effect later on. And I like to look at things broadly. And of course, you're interested in antibiotic resistance, but if your principle works, as it was mentioned for cancer, also herbicides, insecticides, rodenticides, everything are probably following the same principles. And I'm interested in stressors in general, and I sort of look at them as either disruptive or resource limiting. And I'm assuming I think your antibiotics are damaging, disruptive, rather than, I don't think they're protein synthesis inhibitors, which would be resource limiting, is one way to classify them. But I was wondering, you could do that. It would be interesting to see the hysteresis experiment using, say, just any kind of toxin or heat or free radicals or something as one of the, either the before or the after, and see, to see how broad this is. Is it just limited to a couple of antibiotics, or is this any type of stressor at all? And then you've got a real powerful general principle that I'd love to see. I'd love to see advance. Of course, this is a much bigger lab than, but anyway, think big, I guess is what I'm saying.
No, definitely a very good point. I would phrase it differently. I think what is neglected, or where we miss data, is on transient effects that could influence antibiotic efficacy. So most research that's done by evolutionary biologists is focused on looking at genetic changes, obviously, because that is evolution. But to what extent inducible physiological effects play a role, that is somewhat neglected. We have done, in our most recent work, we have tested a couple of other stressors. We have not really done, it depends on what you call toxins. But the result we have so far is that this phenomenon, as we describe it at the moment, requires that you have some disruptor of membrane integrity. And in these gram-negative bacteria, particularly the inner membrane. This is what we can say so far on the additional compounds that we used. But it has clearly not been exhaustive, and I agree with you, it would be actually very fascinating, exciting to see a much broader test of this. Also briefly wanted to comment, because it's a 15 minutes, this is actually very important to prove that there is this inducible physiological effect, and to be certain that this is not due to selection happening within one generation. So we wanted to really make sure that it is an inducible effect. But of course, the effect also occurs if you have exposure to the first stressor for a longer time period. So it's simply the experimental setup to prove that this is based on a physiological effect.
Okay, thank you very much. We are at the top of the hour and we have to close out. We're starting to see a drop off in numbers as people go to their next meetings. Aditi and Hinrich, thank you so much for a fantastic talk. It was really great to have you bring this work to life so much and to hear the great questions. There are a lot of great questions still left in the chat. I wish we could get to them, but they will be preserved for you in the recording, and you can look through those and perhaps respond to some of the people directly. Before we close out, I want to just share with you the next two upcoming Club EvMeds. Hopefully I'm sharing my screen here. Here's the great talk we just heard. And for those of you interested in infectious disease, we have two great speakers coming up. We have Troy Day on April 7th at noon Eastern time. And he'll talk about the evolutionary epidemiology of infectious diseases. Should be very interesting. And then after that, we have Jess Metcalf from Princeton, April 25th, again at noon Eastern time. And we don't have a title yet from Jess, but I'm sure it'll be a great talk as well. So with that, I'll thank our speakers again, and Meredith, of course, for all of her help, and thank all of you for tuning in. Hope to see you at the next one.